I'm Kate Meurs. I've put together some slides. Many of these are the same that I showed when I went to the national meeting in October. But one of the best things about this type of technology that both Cheryl and I have found and really enjoy is that it really offers us an opportunity to have almost one-on-one conversations with you about this disease and about questions that you might have. As you may or may not know, dilated cardiomyopathy is just a primary muscle disease of the heart. It's not usually caused by low thyroid or necessarily nutritional things, but we know it is inherited, at least in some breeds, and it's definitely inherited in the Doberman Pinscher. In the United States, the Doberman Pinscher is the most commonly affected breed of dog with it, and affected dogs die of sudden death, so they may be out running or they may just be sitting next to you on the sofa one night and fall over dead as if they've had a heart attack. More commonly in the Doberman, what happens is their heart really begins to fail, so they become short of breath, they start to cough, they lose weight, they become very tired, and unfortunately, eventually, many people have to eventually put them to sleep because the medication no longer keeps them comfortable. What is important about this disease when it comes to the dog is that it is different in every breed of dog. So if you have a friend who has Great Danes and they also get dilated cardiomyopathy, or St. Bernards, this is not the same disease. Dilated cardiomyopathy in the Doberman Pinscher is a very unique disease specific to this breed. And these are just some echocardiograms that I've shown many of you before. This one up here is the normal dog. So this is the normal dog up here, and this is the left side of their heart, the heart that is usually affected with the disease. This is the abnormal dog, the dog that has dilated cardiomyopathy. Here is their left ventricle. And you can see how much bigger this chamber is in comparison to the normal dog. That's the dilation that we talk of associated with this disease. Now, when that happens, when the heart gets big and dilated, its ability to function also severely decreases. And it disrupts the electrical system of the heart, and that's why many of these dogs die suddenly and have a heart attack. These are the electrical abnormalities. This is a normal dog's electrocardiogram. It's also sometimes called an ECG or an EKG. And you see, in a very normal dog, there should be a small spike followed by a very tall spike, and they should be quite regularly spaced. Here's the affected Doberman, and you can see it sometimes has a small spike followed by a tall spike. But then here's another normal beat, and there are two very close together beats that follow it, and then he goes back to having three normal beats again and a few abnormal. It's these abnormal ones that can lead to electrical defibrillation and sudden death. So I have one question, and that's excellent. The first question is, what is the evidence that sudden death form is the same as the congestive heart failure form? This person had a dog with sudden death with only borderline structural changes. That's a really good question. I don't know that there is evidence. We know from studies that have been done by Dr. Calvert at the University of Georgia, I'm sure many of you worked with him over the years, he's studied this disease for a long time, and he reported that about 30% of Dobermans with heart disease that he saw, adult Dobermans, had these ventricular premature beats, these abnormal beats here, and were at risk of sudden death. And we made the assumption, based on his information, that he saw a lot of Dobermans that had these abnormal beats and sudden death, and that it was seen in Dobermans, and that they were generally middle-aged. We made the veterinary profession made the assumption that that was the same disease, that cardiomyopathy in some families of dogs perhaps, or in some dogs, we don't even know if they're distinct families, had these abnormal heartbeats and were at risk of sudden death or fainting, whereas others were more likely to show the weakened heart muscle. So we basically made the assumption that it's one disease. I once upon a time tried looking at it in families, and what I did find was that there were some families that did tend to have more of the sudden death, and some families certainly that we see much more of the heart failure. And so I agree with you. I am not convinced that this is the same disease. The dogs in which we found the genetic mutation were dogs that had the classic dilation and decreased function. So when I set that study up, I specifically echoed those dogs with an ultrasound every year and holtered them with a 24-hour Holter monitor. Some of them also had some abnormal heartbeats, but they were specifically chosen for the study based on dilation and poor systolic function. So I think it's a really good question, and I think in my heart it may very well be a totally different disease. Next question is, we're learning if dogs with diagnosed dilated cardiomyopathy, yet they are negative for the gene mutation you found. Is it safe to say now that there are definitely other genes or mutations that cause the dilation portion of dilated cardiomyopathy? Are there likely other genes? I think there are likely other genes. You have to be a little bit careful when we interpret that data. So in my heart, I do believe there are other genes. If we look at human beings, there are 24 different genes now in which mutations have been found that lead to the development of dilated cardiomyopathy. When I've looked at samples submitted by boarded veterinary cardiologists, so I have the information there that says, here's the dog and this is his echo, I have absolutely seen a very small number, I think about 10 dogs now, where I have seen what is good evidence that they actually have dilation and do not have this mutation. For research purposes, I would greatly appreciate if somebody has those dogs that they let us know, because if we've tested them and they're negative, it's very helpful for us to pull those DNA samples and go back and look at them for other genes. And so far, I've worked with Dr. Estrada at the University of Florida and Dr. Mark Oyama at the University of Pennsylvania, and between us, we've really only gathered, as I said, about 6 or maybe 8 samples that fit that. 8 may be a little high, but we are specifically trying to save those samples so we can go back and look at them. So, yes, I do believe that we have a small number of dogs so far that don't have this mutation. I'm not suggesting that there aren't more of them, but I've not actually seen very many of those. So if you have that information, if you have a dog that was evaluated by a cardiologist and has the disease but is genetic mutation negative, please let me know, because that's very helpful information for us to try to identify another gene. We know that this disease is inherited in an autosomal dominant fashion. So this is a pedigree that I've shown, again, some of you before. I apologize if you've seen some of these slides before. It's just because they're very good illustrations. So in this pedigree, these are multiple generations. So this is one generation. The next horizontal line is a second generation, third generation, fourth generation. This family way up here, this generation way up here, those dogs have a U in them because we don't know their disease status. Black symbols are dogs that definitely had echocardiographic diagnosis of dilated cardiomyopathy. White ones are dogs that were at least 10 years of age and had not developed changes on an echo or on a Holter monitor as having at least 50 VPCs. These are dogs that had normal echoes and less than 50 VPCs in 24 hours. So this is an unaffected dog. So in this generation, circles are females, squares are males. So here's a affected female bred to an unaffected male, and this dog eventually died, and we did a necropsy and demonstrated that he did not have disease. They had, I think, 7 or 8 children. 2 were affected, a male and a female. That female went on to produce an affected son. He produced an affected son when bred to an affected female. And so what this shows you is that the disease goes down every generation, and equal numbers of females and males are affected. So that tells you that it is autosomal, not carried on a sex chromosome, and it is a dominant trait. I have another question that says, as I mentioned, there are dogs with dilated cardiomyopathy that tested negative for my test. There's a dog in Finland that has tested homozygous positive, but is 10 and a half years old. Is this of interest to you, and what do you think explains his longevity? Excellent question. Yes, that sort of information is also very helpful to us. The thing about this disease, and we know the most about it from the research done in human beings, but also a little bit from what we've learned about genetics of heart disease in cats and boxer dogs, is that genetic diseases have what's called variable penetrance. And that means that you can have the genetic mutation for the disease, but how much that disease penetrates that individual animal or the individual human being is quite variable. Some individuals within a family, within the exact same family or within the same litter, will have the mutation and have very severe disease. Others will have the mutation and never develop the full-blown disease. In human beings, again, the species in which the most is known about this, they do not know what affects and what causes the variability in penetrance. Possibilities would be other genes, so genetic modifiers. Perhaps one person, two siblings in a family might even be twins, human beings and twins, and they both inherited the mutation, but perhaps one of the twins also inherited a protecting gene from their mother, and the other twin didn't get that protecting gene from his mother. So there may be genetic modifiers. But other things certainly, and I think this may be true in the Doberman as well, may be things like environment, diet, how much an individual exercises, possibly even things like pollutants. We know that certain things in the environment, like pollutants that come from steel factories or from factories that use a lot of blue-green dye, contain toxins that actually can cause genetic mutations. And so it might be that there is certainly environment there that affects the penetration of that mutation within an individual. And so what we don't know yet is, nobody knows, not even again in humans, how much penetrance is inherited. So we hope that a family that has the mutation, has the disease, but doesn't show it very much, like this dog that the individual asked me about, that dog may have a low penetrant form. And so he might be a dog that could possibly be used for breeding, particularly if bred to a negative dog. How can this be a dominant trait when two carriers had unaffected offspring? Oh, great question. So in this example, so here we have two affected parents, one affected offspring, and two unaffected offspring. So if it's dominant, how does that happen? Well, these are most likely heterozygous. So let's imagine that this dog had one good gene, the capital A, and one bad gene, a little a. And that this dog, the mother, had one good gene, big A. Oh, we'll just make it easier. We'll put B for bad. And this dog has A, and a B. So, in a dominant trait, this male has A, which is a good gene, and B for bad. This female has A for good and B for bad. In a dominant trait, if you just have one copy of the bad gene, you show it. So this dog has one bad B, and he shows the disease. This dog has one bad gene, the B, and she shows the trait. Now, when you breed them, this dog down here could be either BB, he got the bad gene from both his mother and his father, or he could be AB, just one bad copy of it, and he shows it. But these two girls are probably AA. They were lucky, and they got this good gene from both parents, and so they are AA, two good genes. Now, sometimes people write to me and say, well, I thought when you bred two heterozygous, you were going to get 25% negative, 75% positive because of, experimentation with Mendel's, those old arguments. That's for an individual. Every time a puppy comes together, when these two dogs are bred,